So, hi everyone, and thanks for logging into today, which is actually our fifth lockdown webinar series. And this one is entitled Introduction to Histology with me, Emma. So I know for some of you who have been joining us as our lockdown webinars and been going along know this already, but for those of you who don't, our life science team here in MSc is headed by Dr. Lisa Tang, and she did her PhD in the University of Leeds and has about 7 years postdoc experience before she came and worked with MSc. Her main techniques were molecular biology, protein expression, and Western blotting. Then there is my colleague, Dr. Paul Lohan, who did his PhD in NUI Galway. He has about 3 years postdoc experience before he joined MSc, and his main techniques were flow cytometry, cell culture analyses. And then there's me, Emma. I did my PhD in DCU, and I have more than 2.5 years experience postdoc in Trinity, and I did about 6 months of next generation sequencing experience before I joined MSc. But my main techniques really during my postdoc time and PhD were cytochemistry, histochemistry, flow cytometry, and Western blotting. So I'm just going to summarize what I'm going to go through a little bit later today, and then you can feel free to ask any questions at any point. So I'm going to go through what is histology, who does histology. I'm going to do a walkthrough example of paraffin histology because it's one of the more commonly used kinds of histology that gets done. So this involves everything from fixing your samples to processing them, embedding them, sectioning them and mounting them, and then staining them. I'm also going to hit on 2 other brief, small, well, they're not small, but considerations. So there's another type to paraffin histology called cryo-histology, and there's also another kind of, well, staining mechanism. So I'm going to go through really chemical dyes, but I'll also briefly touch on immunohistochemistry as well. And then we can have our question and answer session at the very end. So just to define what histology is, so it's the study of microscopic anatomy or microanatomy of cells and tissues, and it actually comes from the Greek word histos, meaning tissues or columns, and logica, being study. So actually, coincidentally, it seems a bit weird, but in England and in Italy, there were 2 events where Robert Hooke in 1665 started drawing the cellular structure of cork and a sprig of sensitive plant micrographia. And in the same year, Marcello in Italy was drawing lung capillaries of frogs. And you can see they start off very basic drawings, but the histology of today is far more complex. We've come through in leaps and bounds. And not only do we just do single stains right now, you can do a whole multitude of combination stains to result in some incredible images that are both structurally informative as well as incredibly pretty. So who does histology? Well, there's obviously you guys there in research. So you research disease pathologies and do drug and biomedical device discovery. So you're asking questions like, does the tissue or organ change structurally over the course of a disease? Or can my drug or medical device prevent any structural changes? There's those in the clinical world who do disease identification and cause of death identification. So they tend to be asking, is there any structural changes observed to clinically diagnose X disease? So, a very common one obviously is cancer. And were there any infective agents involved? So did they find any bacteria or were there any parasites found in the tissue? And of course, we have our beloved CSI. Anybody who's ever watched the TV series will probably already know, but you can establish cause of death. You can identify unique evidence. So asking questions like, was the death instantaneous or not? Or what trauma was or is observed? So if we have a look just generally overviewing of the process, it looks, how do we get from having this chunk of organ to our pictures? Well, it looks something like this. So you start off with having tissue, then you need to preserve it to stop it going rotten. Then you need to structurally support it and put it in a matrix that allows you to cut it into thin slices that you can then stain. So it looks again something like this, would have a little added step in here. So we fix the tissue in order to prevent decay and degradation. We process the tissue in order to prevent excessive alteration of the tissue during the embedding process. The embedding then contains the sample within that supporting material, which then you can section, which is where you cut the tissue into thin layers and mount them onto slides that you can stain to enhance the visibility of the different structures. So if we start going through these steps one by one and we look at fixing first. So the aim of fixation is to prevent decay and degradation. And this is achieved by using fixative reagents that cross-link protein molecules. And this looks something like this, where you get this binding events between the protein molecules that help prevent your sample rotting. So there's lots of different types of fixatives, and a very, very commonly used one would be 10% neutral buffered formalin. And sometimes you'll notice that actually, so formalin is actually a liquid formaldehyde gas, or it's heat dissolved paraformaldehyde, because sometimes you see people fixing their samples in PFA. You can also get other compounds such as this Debesco-Brazil, so that's really great for renal biopsies. And of course, formalin is toxic, so there have been now non-toxic, non-carcinogenic reagents being brought onto the market very successfully, such as GreenFix, and these are great alternatives if you're looking to move away from toxic chemicals. So how do we go about fixing them? So first, obviously, you're going to place your excise sample into your histology cassettes, and they look like this. You may need to add paper or foam pillows to prevent your tissue from getting distorted within it, or tissue torsion as it's known. You're going to close your cassettes and immerse them in fixative. Now, you may need to trial your fixative depending on what you're trying to do, but a general rule or guide is something like this, where the length of time for fixation is approximately 1 millimeter per hour. And so typically people do protocols that are like overnight for at room temperature, or 24 hours at 4 degrees or in your fridge. The volume of fixative is actually incredibly important, because you need to have more than 15 to 20 times the volume of tissue. And large volumes ensure optimal fixation and fixative stability, because actually fixatives are very poor buffers, and the pH tends to change during the course of the fixation process. So by having excessive volume will help with this. So I've inserted actually a step in here. It's relatively niche, but it's called decalcification. So if you're looking at bones, bones are obviously incredibly hard, and you're talking about putting these into wax, which is very soft. So you need to decalcify very heavily calcified samples in order to cut through and get a really nice clean slice of a paraffin section. So this is typically carried out straight after sample fixation and prior to routine processing in paraffin. So not doing so will actually damage your blade that you're using, so you'd end up going through a lot of blades, and also waste your samples, because you end up with disastrous ribbons. So when you're cutting your samples, they get cut into these really lovely fine thin what's called ribbons, and obviously if you're going to be hitting bone off that is harder than the paraffin, it's going to start ripping your samples to shreds, and you're just not going to get anything that you can put onto a slide. Of course, there are chemicals out there on the market that you can use that are great for just incubating your sample in and doing the decalcification, and obviously protocols vary on how long you need to keep your sample in. If you've got a very calcified sample, you'd obviously need to leave it in there a little bit longer, but there's ones that have been optimized. So this micro DFAST is great for medium-sized samples and compact bone, but there's obviously more specialized ones like bone marrow ones that you can use too. So let's just have a look through our walkthrough again. So we've covered fixing, so now we can move on to processing. So the aim of the process is to put your tissue into supportive medium to allow for thin sections to be produced that can be stained. So we achieve this by infiltrating the tissue with suitable histological wax, in the case of paraffin, of course. So this process involves, firstly, you need to remove the water, and this is done by adding ethanol or alcohol in a process known as dehydration. Secondly, you need to displace this ethanol or alcohol with xylene or a xylene substitute, and this process is known as clearing. And then you need to displace your xylene with paraffin wax, which is a process known as wax infiltration. And these can be done either manually or automatically. So manually, obviously, you're going to have to use containers with these chemicals and you'll hand process them. And if you're fortunate enough to have a tissue processor in your department, they are wonderful pieces of machinery. I've done both, and tissue processors save you a hell of a lot of time. And there's examples of this like the Diapat Donatello, but there's lots of other ones out there on the market. So we'll just go into these in a little bit more detail. If we look at dehydration, so paraffin wax is hydrophobic and it's immiscible in water. So therefore you need to remove the water from your samples before you can start considering putting it into paraffin. You do this using a series of ethanol or alcohol solutions of increasing concentration. And anybody who's had a good night on the town will know nothing better to dehydrate you than a good old alcohol. So the typical dehydration sequence for samples that are approximately 4 millimeters thick would be something like this, where you gradually increase the amount of ethanol and then you do a series of washes in fresh, clean ethanol so that you can make sure that you have full penetration. Next, if we move on to clearing. So paraffin wax and ethanol are also largely immiscible. So you need to remove the ethanol. And you use clearing agents such as xylene. So many, but not all clearing agents impart an optical clarity or transparency due to their relatively high refractive index. So some of them will basically just remove all coloration like this. So clearing agents can also remove fat from tissue because obviously fat creates a barrier to your wax being infiltrated into the samples. And a typical clearing sequence for samples of approximately the same size, like we spoke about before, of 4 millimeters thick, would be something like this, where you do 3 incubation phases with xylene for different periods of time. So it's worth knowing here that actually xylene is another of those toxic chemicals. So what has been really great is that there's moving towards nowadays using less toxic or non-toxic. So there's a great example of this called an Otix reagent, and it has been shown to actually create just as good, if not better, images, which eradicates that whole worry that if you're substituting a really great chemical that might be toxic, but you want to move towards something that's just as good, if not better, and not toxic. So next we're moving on to wax infiltration. So wax needs to be hot, obviously, to infiltrate, because if it's cold, it's a solid. And when you do your wax infiltration sequence for samples of approximately the same size, it looks again something like this. So very repetitive, of course, but you want to make sure that the reason why it repeats itself in that manner is that you want to have clean, fresh wax going in. So we're moving fairly quickly through our processes now, and we're going on to embedding. So the aim of embedding is to create a block that can be cut into thin slices for staining. And this is achieved by placing your wax infiltrated tissue into a mold and filling it with more wax. This is also known as actually blocking out, because the specimen is set in a block of wax. And you have 2 really main requirements for this. You need metal or plastic molds, and these molds come in all shapes and sizes for whatever tissue type you need. And you also need an embedding module, and these embedding modules dispense molten wax, and you need a hot and a cold place to set and remelt or reform your wax as you're trying to embed them. So it goes something like this, where you pour a little bit of molten wax in the bottom of your tissue mold and you place your specimen in the mold, and you allow that to set in place, and you can cool and flatten as required, but you can also heat up again if you need to move your samples around a little bit. Next, you're going to cover it with your cassette, and of course your cassette is the thing that you'll have labeled, so you can keep track of what sample you're handling and using. And then you fill that with wax and you allow that to set, and it'll look something like this. And then finally, you remove your block from its mold to inspect for any issues, and you can store it in the mold at room temperature to protect it from getting chipped or damaged or anything like this. And this is a great phase because you can actually observe if your embedding went really well or not. And if you've got any anomalies in it, all you have to do is simply remelt and redo the whole process, and there's been no damage at all. So we've moved on now to looking at our sectioning and mounting next. So our aim is to cut our tissue into thin slices and put it onto glass slides for staining and visualization. So this is achieved obviously using a microtome and special blades to cut really thin sections called ribbons, like I mentioned before. And then you're going to use a water bath or a hot plate to melt wax sections onto your glass. So this is a picture of a microtome. There's many like them. And then this is just a water bath. So you want to use a water bath that can be placed nearby you because you don't want your little ribbons flying off into the distance. You also don't want it too deep, and you want it nice and clean so that you can get your samples and not have any sort of artifacts on them. You need a few other little bits and pieces. You need things like tweezers and small brushes, scissors, scalpel blades, and microscope slides, of course, because you need those that you're going to put them onto to visualize in the end. So firstly, how we do this is we load our block into the microtome, and then you use special blades to cut them into the thin ribbons. Now, typically you cut sections about 3 micrometers thick, but it really can be anywhere between 1 and 10 micrometers, depending on what you're looking for. And it looks something like this. As the block comes in contact with the blade, you'll get these nice, really thin ribbons. So, must give a word of warning here. Sectioning is sometimes and mostly easier said than done. Because if you've got things like plastic or maybe metal that might be part of medical devices or something like this in it, obviously when you're cutting it, the blade is going to come in contact with that and it can rip. So you might have to optimize the thickness and maybe the processing of your sample for those scenarios. So then what you're going to do is you're going to transfer those ribbons that you made, right? So you're going to transfer them to your clean heated water bath, or again, if you're going to put them onto a hot plate, you just put them on your slide and put them onto the hot plate to melt it on top. And once the wax is melted, tissue can be scooped onto your microscope slides and set aside to dry. And obviously, if this part is not done right, your samples will fall off during the staining phase. So we've moved on now to our staining, which is to enhance or visualize the structures of interest. So this is achieved by using various stains. Now, before you can actually stain, remember your samples are in wax. So you need to remove the wax, which is a process called deparaffinization, and you need to rehydrate your samples before you can start staining them. So an example protocol is something like this, where we have to remove the wax by putting it in your xylene or your xylene substitute. So that will get rid of the wax. And then you slowly rehydrate it by putting your samples into decreasing concentrations of ethanol. And then you'll finally rinse with a bit of distilled water to clean all that ethanol off. And then you'll put it into something like PBS or Hanks for a period of time in order to nicely rehydrate your samples. So when we go to staining now, there are many, many different stains and combinations. So these are just a few of the stains that are currently available on the market, but there are lots. And there's also now you can get kits that combine these things together. So looking at this, you can see that there's a lot of similarities between some of the stains, but the thing is that you can pretty much have these all tailored for different reasons. If you want a multiplex or you need to look at only a few things, it's very much up to you, and it's great. If you've got any questions, feel free to come to us. If you've got new tissue or you're looking for a new staining process, please contact us and we can help you figure out what you'd like to do. So I'm just going to go through some of the really common stains though that everybody uses. So this first one is called hematoxin eosin, and it's commonly just referred to as H&E. So this is actually the most widely used stain in histology, and it allows for localization of nuclei and extracellular proteins. And so you can see in the picture here that actually this staining works on the basis of basophilic structures and acidophilic structures. So basophilic being basic, like nuclei, DNA, RNA, they will all stain blue. And then your acidophilic structures, like your cytoplasm, will all stain red, or like variations of red depending on how acidophilic it is. Next example here is just oil red O. So this picture here is counterstained with Mayer hematoxylin so that you can see the nuclei. So this is obviously for the detection of nuclei and lipids, actually. So you can see here the lipids are in red, and you've got big lipid vesicles there in amongst your cells that are stained blue there. Next, we're going to look at Mason's trichrome stain. So actually, this stain and the next stain, Gieson's trichromatic stain, they stain very similar things, but with a slight difference. And this is kind of what I was saying earlier on, that you can, while they appear to stain the same things, they can be slightly different. So these are both for the differentiation between collagen and smooth muscle. So you can see in the Masons and the Giesons that both the nuclei and the collagen are similar colors. So their nuclei are black, collagen is red. However, the differentiation between these two stains is that in the Masons, your erythrocytes are yellow and orange, and your muscle fibers and your keratin and your cytoplasm are bright red, whereas in the Gieson's trichromatic stain, the cytoplasm, muscle, epithelium, and erythrocytes are all yellow. So it's a great differentiator, and it really does just depend on what you're looking for. Next, if we have a look of Congo red stain, so this allows for the detection of amyloid, and of course amyloidosis is a big problem for both clinically and diagnostically. So you want to be looking for that in like the liver and the brain. So in these sections here, we can see the nuclei is colored blue or violet, and amyloid is in pink or red. And then just to be a little bit completely different, so I know we were talking about tissues there and proteins endogenous to tissues, but gram stains are obviously for bacteria, and you need to sometimes look for them. So this allows for the differentiation between a gram positive and gram negative bacteria. I'm sure you might have all done this at some point in college. It's a very common experiment done very early in your college days. So in this one, your gram positive bacteria are blue or violet, and your gram negative bacteria are magenta or like pinky purple. So that is pretty much the process of paraffin histology. And of course, like everything in science and everything in general, there are advantages and disadvantages to this technique. So the advantages for paraffin histology are that your samples can be stored for multiple years at room temperature and are easy to handle without damaging. And this is a great technique, actually, if you want to preserve your tissue morphology and have a look of your tissue morphology. The disadvantages of this technique are overfixing can mask epitopes. However, I'll talk about it in a little bit, but there are ways around this, and I did it all the time, actually, so it's not even a big deal. You can circumnavigate this, and it's a lengthy process. And so, as I mentioned earlier on, if you've got an automatic tissue processor, this can help greatly with it and allow you to do other things while you're waiting on your samples to be done. So I'd be remiss, like I said, in mentioning that there is a second method to paraffin, and this is a cryo method. So this involves using a compound known as OCT, or optimal cutting temperature. And this is an inert support medium that you use instead of paraffin wax. In this case, though, the samples are frozen and can be embedded prior to fixation, unlike paraffin method. And again, your advantages and disadvantages of this. So the advantages of cryo method is it preserves your enzyme functionality and antigenicity, and you can snap freeze in order to detect post-translational modifications such as phosphorylation. So it really does depend on what you want to do. This method, obviously, you need a different piece of machinery because everything needs to be cold while you're doing it. So with that in mind, if you have a look at the disadvantages, the storage is really not as convenient because you need to keep it for like a year at -80, or you can store it longer at -190 degrees Celsius. Can have formation of ice crystals and may negatively affect your tissue structure if your tissues are not frozen rapidly. This is known as a process as the Swiss cheese effect. I'm sure loads of you have heard of it before, but it's basically where ice crystals form and blast holes in your samples. The sections in cryo are also often thicker than paraffin sections. So this can have an impact really where you get lower resolution and poorer images. So it really does depend on kind of what you're looking for. And considering the fact that it preserves your enzyme functionality, you may actually need to block active endogenous enzymes to stop them from degrading your samples. So a second consideration is immunohistochemistry. So this is done instead of staining with a chemical dye. And this is really a huge topic in itself. So we can host another session on that maybe sometime. But this uses antibodies instead of stains in order to assess your tissue sections. And this allows for high resolution distribution analysis and localization of specific cellular components. So you can do this one of two ways. You can do it fluorescently using fluorophore label antibodies, such as this one here that has a fluorescent dye actually called DAPI, and a fluorescently labeled antibody called RFP, so red fluorescent protein. You can also do colorimetric and chromogenic methods. So this is using enzyme-linked antibodies like horseradish peroxidase, where you combine it with a substrate and you get a color being formed. So obviously the colorimetric ones, you can only do one or two things in it. If you're going to, you can basically dye it with a stain, and then you can do your colorimetric. But obviously fluorescent, you can multiplex, so you can have many different colors as long as your microscope has the capability. And obviously you want to make sure that you don't have your fluorophores overlapping, that you can't detect what you're looking for. So remember earlier on, I mentioned that there was the disadvantage of overfixing can mask your epitopes. So this is really important in immunohistochemistry, obviously, because if you've masked your epitopes, your antibodies can't bind for you to assess them. But there is a protocol basically where you do antigen retrieval, and this can unmask your epitopes. And it depends on really what you're doing, but I used to have to do this all the time for my samples when I was doing immuno. And it was not a big deal. So there's 4 core methods really to doing it. You have chemical, so you can use things like sodium citrate buffers or 1 millimolar EDTA or tris EDTA. You can use heat, so it's very common to have labs have pressure cookers or steamers, vegetable steamers or rice cookers. These are great options. I've put in scientific microwave there because the ones you buy for your households don't have uniform heat sometimes. So obviously when you're processing your samples, you want the heat to be uniform. So it would be best to maybe, if you're going to go with that method, try and get a scientific microwave. And then, of course, there's a combination where you can use the chemical and the heat. So, for example, this is the protocol that I used to do, where I would essentially boil between 80 and 100 degrees Celsius my samples in sodium citrate buffer, and that used to work a real treat. And of course, lastly, there is enzymatic. So you can use things like trypsin in order to unmask your epitopes of interest. So that's pretty much it for today. Just to summarize what we went through, what is histology, who does it? I did a walkthrough example of your paraffin histology, and then gave a brief nod to cryo-histology and immunohistochemistry. And of course, you get a discount for attending, which I'll be sending out soon enough. So we have a great supplier of histology called Diapath, and they're an Italian company, and they are experts on the matter. So please contact us, and we can collaborate with Diapath in order to help you figure out what's best for you. And it's grand if you want to contact us if you're just in the experimental design phase, or even if you're just looking for more advanced stains techniques. And I'd just like to finish by thanking you so much for listening. And of course, if you've got any questions at all, please don't hesitate to contact us, as we'd love to hear from you. And I'll kind of open the floor now, I suppose, to